In this video, we are going to be going over steps 6 through 10 in glycolysis. Understand that in steps 1 through 5, which was discussed in the previous video, those are going to be steps that are energy consuming because we utilize ATP as an energy source for those endergonic steps. On steps 6 through 10 are known as energy generating steps because we will be producing ATP. Let's go through them. So, starting on step 6, remember that by the end of step 5, and just keep in mind, 2 molecules of glyceraldehyde 3-phosphate are the ones that are going to be processed through the next steps. So understand that even though the slides only show one being processed, all of these steps are going to be doubled. So when it comes to step 6, this is an oxidation step. In this oxidation step, as you can see, glyceraldehyde 3-phosphate gets converted into 1,3-bisphosphoglycerate. The enzyme that allows this to happen is going to be glyceraldehyde 3-phosphate dehydrogenase. So, in this process, as you guys can see, we have NAD, a coenzyme, being reduced to NADH. So that means that glyceraldehyde 3-phosphate is going to be oxidized to 1,3-bisphosphoglycerate. So understand that this process is 2 reactions in one. We have a phosphorylation and an oxidation specifically of that glyceraldehyde 3-phosphate. And as I just mentioned, NAD plus gets reduced to NADH. So remember, even though it was not expressed, remember that we're making 2 molecules of NADH in glycolysis, and they happen at step 6. On step 7, we are going to have the first synthesis of ATP. So, as you can see, step 7 is known as substrate level phosphorylation because in this process, we're going to have the enzyme phosphoglycerate kinase. And as you can see, it's going to be utilizing that magnesium, that valent cation, to catalyze this process. Now, here, as you can see, 1,3-bisphosphoglycerate is going to be the source of phosphate. To convert ADP to ATP. So because the substrate is what is providing the phosphate for this phosphorylation step, that's why this is known as substrate level phosphorylation. On step 8, we have an isomerization reaction. And as you can see in this isomerization reaction, the enzyme that is called phosphoglycerol mutase is going to take 3-phosphoglycerate and is going to isomerize it to 2-phosphoglycerate. What we're basically doing in this process is that we are moving our phosphate from carbon 3 to carbon 2. Understand that this mutase enzyme, it is an isomerase and it requires the presence of that divalent cation, in this case is magnesium, in order to make this reaction happen. On step 9, we have a dehydration reaction. Okay, so in this dehydration reaction, as you can see, the enzyme enolase with the use of magnesium cation is going to be converting 2-phosphoglycerate to phosphoenolpyruvate. Now, as you can see, we are going to be removing water and making an enol molecule. The enol motif is this portion of the molecule. Now, understand that in this step, enolase is going to be a lyase because it's going to be removing water from the system, specifically the water comes from the OH here and the hydrogen here combining to make it. In the last step of glycolysis, we have a second substrate level phosphorylation. In this case, we're going to have our substrate phosphoenolpyruvate be our source of phosphate to convert ADP to ATP. As you can see, the enzyme that catalyzes this reaction is going to be pyruvate kinase, and it requires magnesium, as we have seen in other steps in the process of glycolysis, and the result overall is going to be the formation of that pyruvate. Now, as you can see, when it comes to this 10th step is also regulated. Specifically, ATP is going to be inhibiting pyruvate kinase and regulating its activity. So this is an overall view of all 10 steps that we talked about for glycolysis. Understand that I mentioned before that steps 1 through 5 in glycolysis are going to be energy, consuming steps. Because remember that we utilize ATP to phosphorylate our substrate, but also the hydrolysis of ATP generated energy in order for those endergonic reactions to actually proceed. In 6 through 10, they are energy producing steps. And that is because we saw that in steps 7 and 10, we produced 4 ATPs. Now, step 10 is the end of glycolysis. So again, we produce 2 pyruvate molecules. As I expressed before, NADH is also produced, but NADH gets processed in oxidative phosphorylation. And we are going to see that in later chapters. Now, as you can see, when we look at the net reaction, we have glucose converted to 2 pyruvates, but we only see 2 ATPs forming 2 ATPs. That is because even though 4 ATPs are produced overall in the reaction, 2 were consumed. So when you take 4 ATPs minus the 2 that were utilized in step 1 and 3, that's why we have overall net only 2 ATPs in this process. So, let's also summarize the control points of the glycolysis pathway or glycolytic pathway. So I already expressed that there are 3 steps that are irreversible. and that is step 1, step 3, and step 10. Step 1 specifically is going to be inhibited by glucose 6-phosphate. So glucose 6-phosphate is going to be inhibiting hexokinase, and it's not going to allow it to be active. Again, these control mechanisms exist because it depends on the state of the cell. If the cell has enough energy, then instead of taking this molecule and processing it through glycolysis, it's going to be either held or is not going to be processed. So remember, all these regulatory steps exist because it depends on the condition of the cell that decisions are going to be made in order to process glucose and make pyruvate. So step 1 is where the enzyme is inhibited by glucose 6-phosphate. And as you can see, the other irreversible steps, which is step 3 and step 10, are actually inhibited by the molecule of ATP. Remember that ATP is the source of energy of the cell. So in a way, it makes sense that it regulates steps that are irreversible, because at the end of the day, if the cell has enough energy, you want to stop these reactions in the cell pretty high up in the regulatory pathway. So understand that when it comes to the control points for glycolysis, we have regulatory steps on step 1, step 3, and step 10. And they are all regulated by different molecules. Step 1 is inhibited by glucose 6-phosphate, while step 3 and step 10 are going to be inhibited specifically by ATP.